Residual solvents are volatile organic chemicals that are used or produced during the manufacturing of pharmaceutical substances, excipients, or drug products. Although these solvents are generally removed during the manufacturing and drying process, a small quantity may remain in the final pharmaceutical product because some solvents can be toxic. Controlling and testing residual solvent is an important part of pharmaceutical quality control and patient safety. What are residual solvents? Residual solvents are also commonly referred to as organic volatile impurities. They are not intentionally added to the final drug product for therapeutic purpose. Instead, they may remain as residues from the manufacturing process. For example, solvents may be used for reaction, extraction, crystallization, basing, purification, or processing. If the solvent is not completely removed during subsequent processing, a certain amount may remain in the final material. Why are residual solvents important? Residual solvents are important for three major reasons: patient safety, product quality, and regulatory compliance. First, certain solvents can cause toxicity event at relatively low concentrations. Second, excessive solvents levels may affect the quality or stability of the pharmaceutical product. And third, pharmaceutical manufacturers are required to demonstrate that residual solvent levels are within acceptable limits according to applicable regulatory requirements. Sources of residual solvents. Residual solvents can originate from several stages of pharmaceutical manufacturing. They may be introduced during chemical synthesis, extraction, crystallization, purification, granulation, coating, or other manufacturing operation. The actual solvent profile depends on the manufacturing process and the solvent used during production. Therefore, a risk-based assessment of the manufacturing process is important to identify which solvent need to be controlled. ICH Q3C guideline. One of the key international guidelines for residual solvents is ICH Q3C, titled Impurities Guideline for Residual Solvents. The guideline provides a scientific basis for controlling residual solvents in pharmaceutical products. It classifies solvents according to their potential risk and provides limits based primarily on toxicological considerations. Classification of residual solvents. According to ICH Q3C, residual solvents are broadly divided into three classes. Class 1 solvents are solvents that should be avoided. Class 2 solvents are solvents that should be limited because of their inherent toxicity. Class 3 solvents are solvents with low toxic potential and generally have lower safety concerns when used appropriately. There are also solvents with no adequate toxicological data for which additional justification may be required. Class 1 solvents represent the highest concentration. These solvents are known to pose significant risk to human health and should generally be avoided in pharmaceutical manufacturing unless their use is strongly justified. Examples include benzene, carbon tetrachloride, and 1,2-dichloroethane. If a class 1 solvent is used for potentially present, its level must be carefully evaluated and controlled according to the applicable regulatory requirements. Class 2 solvents. Class 2 solvents have significant toxicity and therefore must be limited. Common examples include methanol, acetonitrile, dichloromethane, toluene, and hexane. Each solvent has a specific permitted daily exposure or PDE and corresponding concentration limits depending on the applicable conditions. These limits are stabilized based on toxicological evaluation. Class 3 solvents. Class 3 solvents have relatively low toxic potential. Examples include ethanol, acetone, ethyl acetate, and isopropyl alcohol. Although class 3 solvents have lower toxicity concerns, they should still be appropriately controlled according to the manufacturing process and applicable specifications. PDE, permitted daily exposure. An important concept in residual solvent control is permitted daily exposure, commonly abbreviated as PDE. PDE represents the amount of a solvent that a person can be exposed to every day over a lifetime without an appreciable health risk based on the toxicological assessment. The PDE value is expressed in milligram per day. The concentration limit for a solvent can be derived based on the PDE and the maximum daily dose of the pharmaceutical product. Common residual solvents. Some commonly encountered residual solvent in pharmaceutical manufacturing include methanol, ethanol, acetone, acetonitrile, dichloromethane, isopropyl alcohol, ethyl acetate, toluene, and hexane. The actual solvents that need to be tested depend on the manufacturing process and the solvent used during production. Analytical technique. Headspace gas chromatography (GC-HS). The most commonly used analytical technique for residual solvent testing is gas chromatography with headspace sampling, commonly known as Headspace GC or GC-HS. This technique is particularly suitable because residual solvents are volatile. In headspace analysis, the sample is placed into a sealed vial and heated under controlled conditions. Volatile solvent partition between the sample matrix and the gas phase above the sample, known as the headspace. A portion of this headspace gas is then injected into the gas chromatograph for analysis. Headspace sampling principle. The principle is based on the distribution of volatile compounds between the sample phase and the gas phase. When the vial reaches equilibrium at a specified temperature and time, volatile solvents move into the headspace. The autosampler then withdraw a defined volume of the headspace and transfer it to the GC system. This approach reduced direct injection of the pharmaceutical matrix and helps protect the GC column and system. Gas chromatography separation. Inside the gas chromatograph, the injected compounds are transported by the carrier gas through the chromatographic column. Different compounds interact differently with the stationary phase of the column. As a result, they travel through the column at different rates and are separated based on their retention characteristics. Each solvent produces a chromatographic peak at a characteristic retention time under the specified analytical condition. Detector. After separation, the compound reach the detector. A commonly used detector for the residual solvent analysis is the flame ionization detector or FID. The detector response is converted into an electrical signal and displayed as chromatographic peaks. The peak response is then used for identification and quantitative determination of the residual solvents. Standard and sample preparation. For quantitative analysis, standard and sample solutions are prepared according to the approved analytical method. The standard solution contains known concentration of the target residual solvents. The sample solution contains the pharmaceutical material being tested. Both solutions are transferred into suitable headspace vial and analyzed under the same chromatographic conditions. Identification. Identification of residual solvents is generally based on retention time comparison between the sample and the corresponding standard. When a sample peak appears at a retention time corresponding to a standard solvent, the solvent can be identified provided the method identification criteria are satisfied. Quantification. After identification, the concentration of each residual solvent is determined quantitatively. The response from the sample is compared with the response obtained from the non-standard concentration, depending on the analytical method. Quantitation may be based on external standardization, internal standardization, calibration curves, or other validated approaches. The final result is commonly reported in parts per million or ppm. Simple calculation concept. In a simple external standard approach, the sample concentration can be calculated by comparing the sample response with the standard response while considering the standard concentration and applicable dilution factors. The exact calculation must always follow the approved and validated analytical method. For regulated pharmaceutical testing, analysts should not modify calculation formulas without appropriate scientific and procedural justifications. System suitability. Before evaluating sample results, system suitability requirements must be checked depend on the method. These may include retention time, resolution between critical peaks, peak symmetry or tailing, theoretical plates, and response repeatability or RSD. System suitability demonstrates that the chromatographic system is performing adequately before sample results are expected. Typical chromatogram. A good residual solvent chromatogram should show well-defined and adequately separated peaks. The retention time should be consistent and critical solvents peak should meet the required resolution criteria. Unexpected peak, poor peak shape, inadequate resolution or abnormal response should be investigated according to laboratory procedures. Common problems. Several problems can occur during residual solvent analysis. Common issues include high blank response, carryover, poor resolution, low detector response, high variability, unexpected peaks, and contamination. Potential sources include contaminated solvents or vials, improper sample preparation, leakage from the headspace system, unsuitable incubation conditions, column problems, septum issues, or incorrect instrument parameters. Troubleshooting should be performed systematically and documented according to laboratory procedure. Importance of method validation. The residual solvent analytical method must be appropriately validated before routine use, depending on the applicable guideline and method requirements. Validation may include parameters such as specificity, accuracy, precision, linearity, range, LOD and LOQ, and robustness also. The purpose of validation is to demonstrate that the method is suitable for its intended purpose. Regulatory compliance. Residual solvent testing is an important component of pharmaceutical quality control. Manufacturers must establish appropriate controls based on the manufacturing process, toxicological risk, applicable specification, and relevant regulatory requirements. The analytical procedure, acceptance criteria, calculations, chromatograms, system suitability data, and results should be properly documented and traceable. To summarize, residual solvents are volatile organic compounds that may remain in pharmaceutical materials and manufacturing. Their control is important for patient safety, product quality, and regulatory compliance. ICH Q3C provides an internationally recognized framework for classifying and controlling residual solvents. Headspace gas chromatography or GC-HS is widely used because it is well suited for volatile compounds. Finally, reliable residual solvent testing depends on a suitable validated analytical method, appropriate system suitability, accurate calculations, and proper documentation. Effective control of residual solvents is not simply an analytical requirement, it is an essential part of pharmaceutical quality and patient safety. Why understanding these sources, classification, analytical principle, and regulatory expectation, pharmaceutical professionals can ensure reliable control of residual solvent throughout the product life cycle. If you found this video helpful, share and subscribe.